Dear students, next topic is enzyme kinetics. In this section, we can discuss about mechanism of enzyme action, its regulation, type of enzyme inhibition. Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. Dear students, enzyme kinetic means the rate of an enzymatic reaction. You know, enzymes are biological catalyst or biocatalyst which help to accelerate a biochemical reaction. The basic principle of enzyme catalysis is, enzyme react with the substrate and form enzyme substrate complex. See here, in this reaction, there is a site called active site in the enzymes that we already discussed in our previous video. Active site of enzyme and their properties. Substrate bind with the active site of the enzyme, products will form and enzyme will recover as such after completing a biochemical reaction. See, what is kinetics, rate of enzymatic reaction? Actually, there are different model for the mechanism of this induced fit model, then lock and key hypothesis are the mechanism of enzymatic reactions explained by various scientists. As we already discussed, the substrate is bound in the active site by multiple weak forces which result into the enzyme substrate complex. Originally, there are two models were proposed to explain how an enzyme binds its substrate. First one is the lock and key model and the second one is induced fit model. The lock and key model was proposed by Emil Fischer in 1894. According to this model, the shape of the substrate and the active site of the enzyme are thought to fit together like a key into its lock. The two shapes are considered as rigid and fixed and perfectly complement each other when brought together in the right alignment. See, the figures were shown how the lock and key mechanism and to this enzyme and substrate bind and form enzyme substrate complex. The second one is induced fit model. It was proposed by Daniel E. Koshland in 1958. It states that the binding of substance induces the conformational change in the active site of the enzyme. In addition, the enzyme may distort the substrate, forcing it into a conformation similar to that of the transition state. For example, the binding of glucose to hexokinase induces a conformational change in the structure of the enzyme such that the active site assumes a shape that is complementary to the substrate, that means glucose, only after it has bound to the enzyme. The reality is that different enzymes show features of both model with some complementarity and some conformational change. See here, the first diagram is binding of glucose to hexokinase and the second one is the diagrammatic representation of active site on the enzyme and substrate. So the next is the activation energy. We already discussed about this in previous section of in the classification of enzymes. All the chemical reactions in a biological system have an energy barrier which prevent reactions from proceeding in an uncontrolled and spontaneous manner. The input of energy required to break this energy barrier or to start a reaction is called the activation energy. Enzymes lower the activation energy of a reaction, that is, the required amount of energy needed for a reaction to occur. They do this by binding to the substrate and holding it in a way that allows the reaction to happen more efficiently. Next is coenzyme. Enzymes functional group take part in acid-base reaction to form transient bonds substrate or take part in charge-charge interactions. They are less stable for catalyzing redox reaction or transfer of a chemical group. Such reactions are carried out by small molecules called cofactors. Cofactors may be Metal ions such as Zn2 plus required for the catalytic activity of carboxypeptidase A or organic molecules known as coenzymes such as NAD plus in YADH. Some cofactors, for instance NAD plus, are but transiently associated with a given enzyme molecule so that in effect they function as co-substrates. Other cofactors known as prosthetic groups are essentially permanently associated with their protein, often by covalent bonds. For example, heme prosthetic group of hemoglobin is tightly bound to its protein through extensive hydrophobic and hydrogen bonding interactions together with a covalent bond between the heme Fe2 plus ion and his F8. The enzymatically inactive protein resulting from the removal of a holoenzyme's cofactor is referred to as an apoenzyme. Apoenzyme, that is inactive, plus cofactor give holoenzyme. That means that enzyme will become active. Here some of the example of coenzymes and the reaction catalyzed by that. Then vitamin coenzyme and human deficiency diseases are shown. Then the rate of enzymatic reaction was studied by Michaelis and Menden. That is called Michaelis-Menden's equation. So what is Michael-Menden's equation? See, velocity of or the rate of an enzymatic reaction is equal to Vmax into substrate concentration divided by Km plus S. What is Km? Km is Michael-Menden constant. Michaelis-Menden constant. Next we can see what is Michael-Menden equation in detail. Leonard Michaelis and Maud L. Menden in 1913, while studying the hydrolysis of sucrose catalyzed by the enzyme invertase, proposed this theory. This theory is, however, based on the following assumptions. Only a single substrate and a single product are involved. Process proceeds essentially to completion. Concentration of the substrate is much greater than that of enzyme in the system. An intermediate enzyme substrate complex is formed. The rate of decomposition of the substrate is proportional to the concentration of the enzyme substrate complex. This theory postulates that the enzyme forms a weakly bonded complex enzyme substrate complex with the substrate. This enzyme substrate complex on hydrolysis decomposes to yield the reaction product and the free enzyme. That means E plus S gives ES, that gives E plus P. Then Michaelis-Menten equation is V is equal to Vmax into S divided by Km plus S. Here in Michael-Menden's constants, letters explanation are given. Michael's constant is numerically equal to the substrate concentration, or in other words Km is equal to the concentration of the substrate, which gives half the numerical maximal velocity, that is Vm. It is noteworthy that for any enzyme substrate system, Km has a characteristic value which is independent of the enzyme concentration. Next we can discuss about what is the significance of Km and Vmax. What is Km? Michael-Menden's constant. Michael-Menden constant is Km and Vmax, maximum velocity of a biochemical reaction. Km value is used as a measure of an enzyme's affinity for its substrate. The lower the Km value, higher the enzyme affinity for the substrate and vice versa. Km value also provide an idea of the strength of binding of the substrate to the enzyme molecule. Lower the Km value, more tightly bound the substrate is to the enzyme for the reaction to be catalyzed and vice versa. Km value indicate the lowest concentration of the substrate the enzyme can recognize before reaction catalysis can occur. An enzyme's Km describes the substrate concentration at which half the enzyme active sites are occupied by substrate. Km value is also used as a measure of the substrate concentration when the reaction rate half maximal velocity, that is 50%, or Km is equal to half Vmax. The maximal rate reveals the turnover number of an enzyme, that is the number of substrate molecule being catalyzed per second. This varies considerably from 10 in the case of lysozyme, 600,000 in the case of carbonic anhydrase. Dear students, Next, we can see what are the factors affecting enzymatic activity. There are many environmental factors affect enzymatic activity that you know. The factors are temperature, pH, concentration of substrate and concentration of enzyme. Here, temperature is one of the important factor that affect enzyme activity. Increasing temperature increases the kinetic energy that molecule possess in a fluid. This means that there are more random collision between molecules per unit time. Since enzyme catalyze reactions by randomly colliding with substrate molecule, increasing temperature increases the rate of reaction forming more products. As temperature increases, more enzymes molecules active site shapes will be less complementary to the shape of their substrate and more enzymes will be denatured. This will decrease the rate of a reaction. In conclusion, temperature increases, initially the rate of reaction will increase because of increased kinetic energy. However, the effect of bond breaking will become greater and greater and the rate of reaction will begin to decrease. Here is the effect of increasing temperature on the activity of enzyme is shown in a graphical form. Next factor is pH. Any change in pH above or below the optimum will quickly cause a decrease in the rate of reaction since more of the enzyme molecule will have active sites whose shapes are not, that means at least are less complementary to the shape of their substrate. Here is the concentration is one of the major factor affecting enzyme activity. Changing the concentration of both enzyme and substrate affect the rate of enzyme catalyzed reaction. Changing the concentration of a substance only affect the rate of reaction if it is the limiting factor. As the reaction proceeds, the rate of reaction will decrease since the substrate will get used up. The highest rate of reaction, known as the initial reaction rate, is the maximum reaction rate for an enzyme in an experimental situation. Next is the increasing substrate concentration increases the rate of a reaction. See, that is shown diagrammatically. Rate of reaction and concentration of a substrate. However, after a certain concentration, any increase will have no effect on the rate of reaction since substrate concentration will no longer be the limiting factor. The enzyme will effectively become saturated and will be working at their maximum possible rate. Here is a diagrammatic representation of enzyme concentration with rate of reaction. Increasing enzyme concentration will also increase the rate of a reaction as more enzyme will be colliding with the substrate molecules. Next, enzyme activity may inhibit with the different substances. There are different types of enzymatic inhibition like competitive inhibition, non-competitive inhibition and uncompetitive inhibition. We can see in detail about what are inhibitors and how an enzymatic reaction is inhibited. Enzyme inhibitors are substances which alter the catalytic action of the enzyme and subsequently slow down or in some cases stop catalysis. There are three common types of enzyme inhibition: competitive, non-competitive and substrate inhibition. Competitive inhibition occurs when the substrate and a substance resembling the substrate are both added to the enzyme. A theory called lock and key theory of enzyme catalyst can be used to explain why inhibition occurs. See, it is diagrammatically given in this figure. When an inhibitor resembles the substrate is present, it will compete with the substrate for the position in the enzyme lock. When the inhibitor wins, it gains the lock position but is unable to open the lock. Hence, the observed reaction is slowed down because some of the available enzyme sites are occupied by the inhibitor instead of the substrate. If a similar substance which does not fit the site is present, the enzyme rejects it, accepts the substrate and the reaction proceeds normally. Non-competitive inhibitors are considered to be substances which, when added to the enzyme, alter the enzyme in a way that it cannot accept the substrate. So that is shown in this figure. Substrate inhibition will sometime occur when excessive amount of substrates are present. Addition amount of substrate added to reaction mixture after this point actually decreased the reaction rate. Dear students, I hope you could understand what is enzyme kinetics, its regulation and in various type of inhibition of enzyme action. Thank you for watching. See you in next class with another topic. Don't forget to leave a like and please subscribe my channel.